Hello, hello, and hello again to each and every single one of you. I am Joseph Harris, and I'm from Grand Canyon University. We're in our course entitled Chemistry 530, in other words, entitled Concepts of Biochemistry. Today, the presentation and the lesson, rather, will be on glycolysis, gluconeogenesis, and the role of aldolase gene expression for today. And I'm under the superb direction of Dr. Karma Cook on this fine day, March 6, 2024. As you saw on the previous slide on the welcome, today we're going to be discussing quite often gluconeogenesis and also glycolysis. In other words, the pathways that they consist of. So the learning objectives today are as follows. The learner will be able to explain glycolysis and gluconeogenesis pathways and incorporate how aldolase is involved in glycolysis and gluconeogenesis. Furthermore, our second objective is the learner will be able to denote different isoforms of aldolase and examine how aldolase B expression can be regulated by specific transcription factors. So, as we always do, I'm going to give you a moment to write down on your paper what is the purpose of learning this objective. In addition, I advise you to underline key terms. What are some words that are unfamiliar to you? What are words that are familiar to you? Think about activating your prior knowledge and think about what do you know about these topics. And if there's something that you don't know, what connection can you make that could kind of make sense of it before we really dive into the actual content. So once again, I'm going to allow you a few minutes to decipher, and then we will work in our pairs, and then we'll come back together and discuss certain tactics and certain things that you have displayed. Alrighty, class. Thank you once again for your participation. You all did a phenomenal job. Again, pat yourselves on the back once again. Again, everybody give yourself a clap. I truly enjoyed hearing the engagement, and I truly enjoy the camaraderie that you all had, within each other. And I also really greatly appreciate how you all truly understood the assignment rather and just did a phenomenal job. So great job to each of you. All right, so now that we know our objectives, so now that we know our end goal, again, we're going to begin with the end in mind. So our end, once again, is to measure those objectives. And how are we going to do that? Well, here's our agenda for the day. All right, so we're going to start off with an overview of the pathways of glycolysis. We're also going to discuss the overview, an overview rather, of gluconeogenesis pathways. In addition, we're going to ask ourselves, what exactly is aldolase? How exactly is aldolase involved in glycolysis? How is aldolase involved in gluconeogenesis? And we're also going to discuss other techniques such as isoforms of aldolases and different expressions. And we're also going to discuss exactly what is aldolase B. And we're also going to discuss once again expressions of those. And then we'll have a conclusion at the very end. And alrighty, to all of my gentlemen, it's time to dive on in, and it's our time to shine. It's our time to get this objective in. So everybody, y'all know my words. Let's do it. All right, so I'm gonna start first with an overview of the pathways of glycolysis. And I'm gonna start first with a tad bit of history. All right, so we're gonna know right now glycolysis is a very old pathway. Some say it's ancient. So once again, glycolysis is an old pathway, and it's so old, or ancient rather, that it evolved before the oxygen molecule was even present in the atmosphere of the Earth. And I think that is just so very cool. So once again, glycolysis is an old pathway, and it evolved before the oxygen molecule was ever present within our atmosphere. Furthermore, we're going to note that glycolysis is conserved within us. In other words, well, of course, not as humans, but also, of course, to organisms, to living things. It's also noteworthy to add. Think about the word glycolysis, Lysis. And I overheard someone earlier during your small groups that someone mentioned that Lysis means to break up, or like to break down, or in other words, to split. I'm gonna think about that when I make, of course, my next point. So glycolysis, um, with this process, one molecule of glucose, again, is simply, of course, going to split, and that one molecule is going to split into two molecules of pyruvate. And with it said, and that's going to be the end product simply of glycolysis. So once again, that one molecule of glucose is going to split, what have you, again, LYSIS, the list of glycolysis, and it's going to, of course, turn into pyruvate. And pyruvate is the end specific result or end product, rather simply of glycolysis. Furthermore, in the presence of oxygen, the pyruvate molecule, it will go into the mitochondria of the cell. It's going to be oxidized, of course, to acetyl-CoA. Furthermore, in the absence of oxygen, the pyruvate molecule is going to be reduced into what we know as lactate. Furthermore, something also very interesting, glycolysis will consist of 10 reactions. Yes, once again, 10 reactions. So be sure, of course, to write that down. And also, in glycolysis is going to occur also in the cytosol of the cell. It's going to generate 2 ATP molecules. On the contrary, oxidative phosphorylation is going to generate 30 molecules of ATP. However, this process is going to occur in the presence simply of oxygen. Furthermore, simple sugars, so think carbohydrates to a point. So simple sugars are going to embrace the capability to enter glycolysis, such as fructose, galactose, sucrose, and also others. And furthermore, glycolysis is further known as an exergonic, think outside, an exergonic reaction. Furthermore, glycolysis is the only pathway that's capable of generating ATP in anaerobic conditions. In other words, in the absence of oxygen, or in cells that do not have a mitochondria. For example, erythrocytes. Also, when glycolysis is in the presence of oxygen, the glycolysis pathway will generate pyruvate. Again, we just talked about that one. And the pyruvate is going to ascend into the citric acid cycle, in addition to also the Krebs cycle as well. And furthermore, a large amount simply of metabolites within glycolysis in the citric acid cycle. Again, that they may go into anabolic pathways, and again, they may generate NADPH and also certain fundamental entities simply that's needed, of course, to maintain, of course, to somewhat stimulate processes such as protein synthesis, such as certain biomolecules such as lipids, certain things such as glycogen, and so much more. And next up, we're going to discuss an overview of the pathways of gluconeogenesis. Well, we're going to start off by stating that gluconeogenesis is classified as a process which allows the body to create glucose from non-hexose precursors. Furthermore, in a lot of instances, gluconeogenesis may be referred to as reverse glycolysis. And furthermore, gluconeogenesis may solely occur within specific tissues of the body. Furthermore, in humans, gluconeogenesis occurs in the liver and sometimes in the renal cortex. And furthermore, a specific amount of glucose may be synthesized by gluconeogenesis. However, if gluconeogenesis fails, again, fatality could be a consequence. Furthermore, beginning with the pyruvate molecule within the mitochondria, pyruvate carboxylase is transformed or is changed into oxo acetate. And furthermore, this change, this transformation, is going to reverse the reaction, and that's going to be in a state simply of non-equilibrium within or by the pyruvate kinase. Furthermore, gluconeogenesis is going to involve substrates, and substrates such as glycerol, lactate, and so much more. And furthermore, the reactions simply of gluconeogenesis are endergonic, think inside. So once again, these reactions are endergonic, and they're also reversible. Furthermore, the controls also exist in the process of gluconeogenesis in lots of levels again, and they ensure simply that all things that are associated, what have you, all associations, they progress, and they're going to progress until the molecule of glucose in the free state, rather, is produced. And furthermore, the substance known as glucagon is also involved, and glucagon is prevalent. It's important, and it's a promoter, simply of gluconeogenesis, and also insulin is also an inhibitor of gluconeogenesis as well. Furthermore, someone may be fasting. In other words, someone might not eat for a certain period of time. So if an individual is in a state of fasting during like we can say 18 to 24 hours, during the time period of gluconeogenesis is going to be involved. And it's going to react. It is going to mainly react in the liver simply of the body. And furthermore, gluconeogenesis is going to sustain blood glucose levels and of course the event simply of starvation. All right, so before we dive into more content, we're going to talk quite a bit about aldolase. And this does correspond with our lesson objective. So be sure to scribe this information in. All right, so again, what is aldolase? What we know right now, aldolase is often referred to as fructose 1,6 biphosphate aldolase. And of course, for short, again, lots of scientists and researchers classify this simply as aldolase. Well, aldolase is responsible for catalyzing, or in other words, speeds up a reverse aldose condensation. And also, aldose is going to embrace the function simply to cleave and also to yield to the hydroxyacetone phosphate, which is simply known simply as, of course, an aldose. Furthermore, we're going to note that two classes of aldose they exist, and we're going to talk about those more. So class 1, and that's located in plants and also in animals. And we also have class 2, which is simply located in bacteria and also fungi. Furthermore, class 2 enzymes, again, they're going to form a zinc ion, and it's going to specifically happen, or it's going to happen simply at the active site. And furthermore, the zinc ion simply, of course, this form, of course, in class 2 is also going to coordinate with the carboxyl oxygen as well. And also going to note that aldolase is often used simply as a drug target. And that drug target often inhibits the enzyme to undergo glycolysis and also other processes. So just a couple of tidbits of simply to learn and to know simply about aldolase. And all right, to my ladies and gentlemen. All right. So we discussed the pathways of gluconeogenesis. We discussed the pathways of glycolysis, and we had a brief discussion as to what exactly is aldolase. So our job right now, in order to meet our objective, is to tie all these in together right now. So the question states, and it's on the slide. It says, how is aldolase involved in glycolysis and also gluconeogenesis? And that's a part of our objective, and that was in our agenda as well. So of course we have to know exactly what, of course, entails specifically the pathways of glycolysis, the pathways of gluconeogenesis, and furthermore exactly what is aldolase and how they're going to correspond, how they're going to associate with each other as well. So first things first, once again, how is aldolase involved in glycolysis? How is aldolase involved in gluconeogenesis? And I'm so glad that you asked those two questions because we're about to answer those right now. All right, so aldolase is involved in the processes, once again, of gluconeogenesis and also glycolysis. And we're gonna note also for right now the catalytic ratio that involves gluconeogenesis and also glycolysis is going to display that aldolase B is an efficient isozyme. We're going to note also that aldolase has been researched heavily and will often bind proteins. Again, they may have absolutely nothing to do with glycolysis, and we can reference the F-actin in that persuasion. And furthermore, actin binding proteins, again, aldolase, is going to correlate. Is going to correlate with, of course, the actin with the sector simply in a physiological sense. Due to the corroboration simply of the actin bind or bound aldolase. And as this is going to display as an inhibitor simply of glycolysis respectively. And furthermore, non-spherocytic hemolytic anemia, that's known simply for its deficiencies in um, glycolytic enzymes. And furthermore, erythrocytes, they depend on glycolysis, of course, in order to maintain and sustain energy. And furthermore, aldolase B is well equipped for gluconeogenesis, and it's going to be equipped within the liver of the body. Also going to note that aldolase B is often referred, excuse me, or is often restricted rather in the small intestine, the liver, and also the kidney cortex. And furthermore, we're going to note that glucocorticoids, they're often discharged, or in other words, released upon hypoglycemic stress, in which the hormones are going to maintain what we know as transcription, many genes again that are involved, of course, within the process of gluconeogenesis. And alrighty, so everyone. All right, so possibly one of my favorite slides and one of my favorite things to discuss are going to be isoforms of aldolases, and we're also going to discuss the different expression patterns, respectively, as we go through these at this time. All right, so we're going to note for right now, again, there are three isoforms of class 1 aldolase that currently exist. And they are, as we see on the screen, we have aldolase A, and we also see the abbreviation, and we have aldolase B, and we also have aldolase C, respectively, also, at this way. And we're going to note also these isoforms are displayed in certain areas simply of the body. And we're going to note they have similarities in the catalytic mechanism, and they also embrace similarities, of course, in molecular mass as well. All right. We're going to also note simply that aldo A or aldolase A, excuse me, is going to simply be displayed primarily in the muscle tissue and also in red blood cells. Aldolase B, or as we see, aldo B, respectively, it's going to occur mainly in the liver, small intestine, and also the kidney. Additionally, and furthermore, as we see, aldo C, with the abbreviation for aldolase C, that's going to mainly occur within the neuronal tissue, the brain, and also the smooth muscle. All right, and we're going to see right now again that all three of these aldolases they play parts in glycolysis in their pathways. However, we're going to note a couple of differences. All right, so aldolase C and B, they're going to perform what we're going to refer to as something called like a fructolytic function. So again, that's something, and if we know aldolase A is not involved in that one specifically. We're also going to note right now that class 2 aldolases, they're going to vary. And we're going to think about bacteria as well. And you can think about E. coli is going to share 10% amino acid identity with other bacteria within class 2 simply of the aldolases as well. And furthermore, we're going to note at this time that all three types or all three of these aldolases, respectively, they have high sequence identity. And furthermore, aldolase A and aldolase B, they have 68% of the same identity. And we're also going to note as well that aldolase A and also aldolase C, again, they embrace or they share 78% of the same identity. And those are simply a few specifics for the isoforms of aldolases, respectively, in their different expression patterns. And alrighty, all right. And before we get to a portion that's very like nitty gritty, as we're going to discuss in a moment, HNF1, we're going to discuss HNF3, C, EPB, and we're also going to discuss DBP. And a part of that is going to be what we're going to refer to as aldolase B. So we know a tad bit right now just about aldolase. We know once again fructose 1,6-biphosphate aldolase is its full name. We know, of course, they have the function to cleave and also to yield certain things. We know the two classes of aldolases exist, and we know the class 1, of course, in plants and animals, and we know the class 2 is going to deal with fungi and bacteria. And we also know that aldolase, is often used as a drug target. Well, aldolase B has its set of functions as well, and we're about to discuss those right now. So the course says, what is aldolase B? So aldolase B is a gene, and it's a gene that's transcribed in the small intestine, the kidney, and also the liver, simply at an extreme high level. I was going to note that aldolase B is the only expressed isoform in hepatocytes. It's also located in the small adult intestine in association also with aldolase B and also, of course, or aldolase A. Aldolase B, it has the ability to catalyze. It's going to catalyze a reversible cleavage and also is involved in gluconeogenic and also glycolytic pathways in addition. Furthermore, this gene transcription of aldolase B is, of course, maintained. It's involved simply or sustained by diet and also hormone in that way. And their promoter of simply aldolase B is specific for the liver. However, it's going to mandate the presence simply of an intronic enhancer in order to be fully and strongly active in specific um, certain types of cells. I can say that. So all right, so now that we have some knowledge regarding LSB, our job now is to discuss the expression regulation, of LSB by transcription factors such as HNF1, HNF3, C dash EPB, and also DBP as well in that respect. All right, so once again, LSB, again, that's our focal technically for right now. And LSB is gonna the proximal promoter simply is maintained by 5 entire elements. And we can think about from 5 prime to 3 prime, and we can think about a negative DE element. It's gonna exist. It's gonna activate a C EBP and also a DBP body site as well. In other words, they're gonna have 2 full body sites, and they're gonna overlap, and that's going to consist of HNF1 and also HNF3 in that specific way. Furthermore, experiments they correlate, of course, with LSB and also dash CAT, and they're going to establish expression vectors for a multitude of transcription factors that are carried out in certain hepatoma cells. Furthermore, DBP and also HNF1, they are very, very strong, and they're also known as transactivators. And they're going to be transactivators of the promoters, again, that correspond with Aldolase B and also CEPB, what have you. And those are known as weak activators as well. So, what have you. Again, furthermore, HNF3, it does not modify certain types of activity, respectively, in that point. And again, in deletion of specific native elements, they often result within trans activations by DBP and also by CEBP, and they're also enhanced for the specific latter portion in that way. And also in hepatocytes, the strong activator is known as CEBP, and also in this way, DEBP is also inactive simply at this juncture in time. Alrighty, ladies and gentlemen. All right, so we're nearing the end simply of this presentation, simply of this video. First and foremost, I want to thank you all in advance for your participation and for your eagerness and for your questions. I appreciate you all. All right, so as conclusion, as I conclude our lesson for the day, we're just gonna note right now that glycolysis once again is an old pathway, and once again this occurred before oxygen was even present in our atmosphere. Again, very interesting. Also, we're gonna note that one molecule of glucose was split into 2 molecules of pyruvate, and of course, known as the end product or the result simply of glycolysis. And furthermore, we're going to note that glycolysis is the only pathway that's capable of generating ATP in a specific, respectively, in an anaerobic condition, respectively, in that specific way. Also, going to note that gluconeogenesis, again, it allows the body to create glucose, once again, from non-hexose precursors. And we're also going to note as well as that gluconeogenesis may be referred to as reverse glycolysis and may solely occur within specific tissues. Also going to note also that gluconeogenesis involves substrates such as glycerol and such as lactate. And also going to note that gluconeogenesis is going to sustain blood glucose levels simply if starvation occurs within the body or within anyone. And for more aldolase, of course, a very prevalent topic for today. And of course, referred to once again simply as fructose 1,6-biphosphate aldolase. And of course, people, scientists, researchers say aldolase definitely for short. And furthermore, there are 2 classes simply of aldolase, class 1 and of course class 2. And of course, class 1 mainly of plants and animals, and class 2 of course deals of course with fungi and also bacteria in that way. And once again, aldolase is often simply used simply as a drug target, and often sometimes inhibits the enzyme to undergo the process of glycolysis respectively. And also once again, there are 3 forms simply of isoforms rather assembly of class 1 aldolases, and all 3 classes they're coded of course by 3 different genes. And of course we have aldolase A, aldolase B, and also aldolase C. So allelase A, of course, once again in the red blood cells, in the muscle tissues, and of course allelase B, of course, in the liver, kidney, small intestine, and furthermore allelase C is mainly, of course, going to be in the hormonal, well, not the hormonal, the neuronal tissue, excuse me, often the brain, and often the smooth muscle as well. And of course, all three of those, as we see on the screen, of course, they're also going to have the high sequence identity, respectively, at that point. And the concluding slides are simply just are simply um, just references. So I do thank you all for your cooperation. I thank you for your participation. If you have a question, a concern, or anything, definitely feel free to email me. I will stay after class today. Thank you all so much, and I will see you next time. Thank you so very much for a wonderful class to each and every single one of you. I appreciate you.